Palm Beach is a community that's incredibly philanthropic. Palm Beach is a community that desperately cares about our issue of Alzheimer's and dementia. Palm Beach is a community that likes to bond together to do something, to spend time with its philanthropic power to make a difference. Palm Beach is the perfect place for the Alzheimer's Drug Discovery Foundation to advance its science and increase its philanthropy. Palm Beach cares.
I've been thinking a lot about hope, just in that I'm doing kind of an inherently really optimistic thing right now, which is that I'm pregnant, about to have a kid, and thinking about, the world that they're going to be born into.
Hope is having optimistic, positive expectations.
Hope makes new beginnings possible.
It gives you that motivation and that optimism to keep going. Whatever it is that you're striving for, it lights the fire to get it done and to work together.
Hope is a feeling of expectation and to make things happen, and that is the definition of my husband, Mel Goods. Mel gave his first speech in 2010 at the Hope on the Horizon dinner, and he was very excited about what was happening then, and here we are almost 11 years later, and so many exciting things are happening now.
I don't expect biomedical science to ride to my rescue, but I will go out a happy man if we can change the course of this disease for the hundreds of millions of people who will soon be at risk. All of us in this room can help to make this happen. Let's keep the hope alive with our generosity, our passion, and our spirit. Thank you very much.
Every morning when he gets out of bed and he walks down the hall and I look at his face and he is smiling, that gives me hope. I can never complain about what is happening in my life because my husband has never given up. And that's the hope he's given me.
I'm hopeful that there will be a world without Alzheimer's, a world that would have allowed my mom to continue on this journey through life with me, that would have allowed her to meet her grandson, her first grandson.
Being part of ADDF gives me hope.
The ADDF and the drug research that it is devoted to brings hope to families, even those families where a loved one is in late stages, because Alzheimer's can happen to anybody.
I'm very, very excited about the promising therapies that are just on the horizon to treat people that might otherwise suffer from the disease.
And now being a part of the Young Professionals Committee, it's truly inspiring seeing people in my age group that have whatever connection to Alzheimer's that may be, that we're all working towards a common goal.
ADDF is so unique that Leonard and Ronald are basically footing all of the expenses. So every single penny, dollar, everything that goes into ADDF goes directly to research. That is truly hope on the horizon.
My mother had Alzheimer's, and after she passed, my brother Ronald and I shook hands and said we will work together to find a cure here, to prevent it and cure it. And we have dedicated our lives to that. We're doing this for a passion. The passion is to make sure that our family is safe and make sure that everyone's family is safe. We promised all of our supporters a cure in your lifetime, in our lifetime. I guarantee it. I've got a pen, a sign on the dotted line.
Hope is here. Now, with the help of a blood test, we can diagnose Alzheimer's. Now, with certain phase 2 clinical trials, we know there are treatments that are starting to show improvement in cognition, and they're safe. When I first got to ADDF, hope was on the horizon. But in the last year, hope is here. It's arrived at our doorstep. How fabulous. Welcome to our 4th annual Palm Beach Luncheon, our first and hopefully last virtual event. I'm Mark Roithmere, CEO of the Alzheimer's Drug Discovery Foundation, and sit back and relax because we have a wonderful program of science and celebration. Our symposium, which will begin momentarily, is going to highlight some of our most promising phase 2 clinical trials. We hope you'll stick around because there's going to be a live Q&A, so get your questions ready. And last but not least, we are thrilled to be honoring the one, the only Paula Zahn, media icon, 9-time Emmy Award winner, also daughter, mother, advocate for all those that are touched by Alzheimer's. And, fun fact, she's also an accomplished cellist.
Please welcome an accomplished news person and no less an accomplished musician, Paula Zahn. Paula, come on.
2020 was a breakthrough year for Alzheimer's science. First, we saw the new blood test that can help diagnose Alzheimer's from the biotech C2N, who the ADDF has been funding for years. Next, there were a series of incredibly hopeful phase 2 studies that are starting to show promise. And then, of course, there's the Biogen phase 3 trial, the very trial of the biomarker being used, the Amyvid PET scan, that was totally developed by the ADDF. Today, overwhelmingly, our portfolio is filled with clinical trials, and 75% of those clinical trials are phase 2 trials. That means they've cleared the safety hurdle, and now we're looking to engage targets that improve cognition. What an exciting time. And of course, none of this progress would have been possible without you, our friends, donors, and supporters. I'm proud to tell you that last year was a record-setting year where we gave out over $30 million of scientific investments. And this year, we're on course to hit between $40 to $50 million. We could not make those investments without you. Thank you.
I'm especially grateful to our extraordinary co-chairs, Nancy Goods, Bonnie Lautenberg, and Heidi McWilliams, staples of the Palm Beach community. Our founding chairs, Nancy and Jan Willinger, and our honorary chairs, Leonard and Ronald Lauder. We want to extend our gratitude to our contributors, especially our presenting benefactor, patron, and supporter sponsors. For those of you who have not contributed to the event yet, we hope that you will consider making a donation by texting ADDF to 243725. And remember, because of the generosity of the Lauder family, 100% of your donation goes directly toward the science.
And now we turn to our scientific symposium, where you're going to hear about some incredible progress in the field.
Joining Dr. Howard Fillit are Dr. Anna Pereira from the Icahn School of Medicine at Mount Sinai in New York City, and Dr. Giacomo Cocchi from the Santa Lucia Foundation in Rome. And here's a quick introduction to Drs. Pereira and Cocchi's work.
My name is Anna Pereira. I'm a cognitive neurologist and neuroscientist here at Mount Sinai in New York City. And my lab studies why certain neurons in the brain are more vulnerable to dying Alzheimer's disease. So our goal is to understand molecularly the vulnerability of those circuits, neurocircuits, and those neurons in order to develop novel therapies for Alzheimer's disease. Through the support from ADDF and the Dana Foundation, we conducted a pilot phase 2 a placebo-controlled trial with riluzole, which is a glutamate modulator that is FDA approved for ALS, Lou Gehrig's disease, and in mild Alzheimer's disease patients. And when neurons are sick, they don't uptake much glucose. So it correlates very well with how functional those neurons are. And what we observed, in a nutshell, is that patients who were in the riluzole group had less decline in cerebral metabolism than the placebo group. I'd like to emphasize that all these findings need to be replicated in a much larger sample size for a much longer period of time so that you can better understand both the safety and efficacy of riluzole in the Alzheimer's population. Just wanted to disclose that both Mount Sinai and I have patents related to riluzole as well. So this is a very complicated problem to solve. But I think I'm optimistic because I think for the last 5 years or so, there's been a major shift of a lot of more attention from multiple levels, from philanthropy, from Alzheimer's advocates, from foundations, and from NIH to really come up to really support Alzheimer's disease research. And I think we have made a lot more progress in understanding the neurobiology of Alzheimer's disease, its mechanism, so that you can potentially advance to novel and efficacious treatments for Alzheimer's and other related dementias.
Yeah, I am Giacomo Cocchi. I am a neurologist and professor of physiology in Italy. My research activity is basically devoted to understand the mechanism of connectivity and plasticity in the healthy human brain, and then try to translate these models into the field of neurodegenerative disorders, and especially in Alzheimer's disease. In the past years, we have been interested in better understanding the alterations in cortical plasticity in the brain of patients with Alzheimer's disease. We now started to think how it was possible to improve this mechanism of altered plasticity, and if this improvement could lead to some also clinical effect. So we started to investigate the effects of drugs such as rotigotine. Rotigotine is a drug that is broadly used in patients with Parkinson's disease. And we found that actually administering rotigotine was resulting in a remarkable improvement of this altered mechanism of plasticity that actually were brought back to the normal levels. So on the basis of this neurophysiological evidence, we have been in touch with the Alzheimer's Drug Discovery Foundation, and luckily we got funded to perform a proper phase 2 clinical trial. And the main results that we got is that delivering rotigotine during 6 months was able to improve the cognitive functions related to the frontal lobe activity. And this improvement was also related and supported by an improvement of the autonomies of daily living. So actually these patients were doing better in this test related to the activity of the frontal cortex, and were also more independent than the patients in the placebo arm.
I'm Howard Fillit. I'm the founding executive director and chief science officer for the Alzheimer's Drug Discovery Foundation. I'm very proud and pleased to be able to welcome you to our 4th annual Hope on the Horizon Palm Beach virtual event, the Scientific Symposium. The science we're going to talk about today is very much about clinical trials, which is the main focus of our research portfolio at this time. As many of you know, we've shifted our emphasis from the preclinical phase of drug research, which involves a lot of chemistry and testing molecules in animals, to the clinical phase of drug development, which involves actually testing drugs in human beings and seeing if they actually work and if they're safe. And along those lines, there are 2 big strategies for discovering drugs. One is called developing new chemical entities, new chemistry, getting new patents, and developing new drugs that come to market. And the other is called repurposing. And repurposing is a way in which we have drugs on the market where we know their safety profile, they're approved by the FDA, or in Europe by the European Medicines Agency, or in other parts of the world. And we know their mechanism of action. There's good reason to test drugs that are on the market that have common mechanisms in other diseases like ALS or Parkinson's disease, and could be repurposed or used in people with Alzheimer's disease. The ADDF is funding about 19 repurposing trials at this point. Most of them are in patients with what we call mild cognitive impairment. People are able to still have, very good quality of life, preventing the progression of that to mild dementia, where functional deficits start to really kick in the disease. And as part of expanding and developing our portfolio of multiple repurposed drugs, and we've had several exciting phase 2 results in these exploratory trials, but we want to actually go to the next step and prove that the drugs really work. and then figure out what we call a commercialization strategy, where there would actually be an indication from the FDA that drugs like riluzole and rotigotine are actually approved for specific purposes, like riluzole slowing the rate of the disease decline in Alzheimer's, or rotigotine treating the symptoms of executive dysfunction, would really enable doctors to prescribe the drug in a very comfortable way and have the drug paid for, so there's patient access. Very pleased today to have two of our ADDF-funded investigators who are leaders in the field of clinical trials and repurposing. Anna, let me ask you to go first. We have exciting results from your trial. What are next steps towards both expanding the science and also a commercialization strategy?
I think that the beauty of studies, small studies, well designed, that can give us pilot information to see if we're going to really move on to a phase 3, hundreds of patients, very expensive trials. So to have a smaller trial that has robust biomarker results that can give us some confidence to move to the next level. And I think it's very important to really have a definitive answer in terms of both safety and efficacy through a phase 3 trial, and then go through the FDA stages of analysis and approval, so that this can be given to patients. We are working on these paths with riluzole, both on the immediate release riluzole that we have used. We use a 50 milligrams twice a day dose in our trial, which is the dose that is FDA approved for ALS. Riluzole, it's a drug that is used twice a day, so the potential opportunities here to have a compound that can have an improved molecule from what we have already. Basically having something that can be given once a day, reduce the risk of liver cell toxicity by improving bioavailability, improving the first pass metabolism in the liver. So the beauty about repurposing that I think Howard was mentioning is that those drugs have been used in humans for many years, even though I think you don't have to start from scratch. So we already have some safety data from decades, but you still need to show both efficacy and safety in the new population. But I think the FDA will likely be interested in fast track drugs that could potentially be repurposed, especially because Alzheimer's has a gigantic socioeconomic impact on society. There's no disease-modifying therapy right now, and so we need really to move fast with safe drugs that have evidence of efficacy to be delivered to patients and families who are so much in need of it.
Let me ask Dr. Cocchi. So our next step again is the commercialization strategy. How do we take next steps with this so it's not just a paper, in a medical journal, and then in 6 months everybody forgets about it. We want to be proactive in really developing this molecule together to getting it to market access and patient access. So maybe you could talk a little bit about the strategy with rotigotine, which I think is a little different than the strategy that Anna's pursuing for commercializing riluzole.
In order to make this drug available to Alzheimer's disease patients, we have to confirm that these effects, although promising that we found in a phase 2 trial, can still be evident in a larger multi-center trial. And therefore, obviously, the next step will be to confirm in a well-designed multi-center, multinational trial the efficacy of this strategy.
I just want to say that that ADDF has filed a patent based on what Giacomo was just telling you. And the inventors are Giacomo and his colleague and myself and one of my colleagues at the ADDF, Dr. Travaglia. And it's the first step in ADDF playing a very proactive role in helping to capitalize on the results from these phase 2 trials, particularly the repurposing trials, in a very meaningful way. To help advance this, relying solely on the universities to do this when they have so many other priorities might not be a very efficient way to help commercialize drugs. And so it's an evolving strategy of ADDF to play a very proactive role in capitalizing on the positive phase 2 results to not only fund the next step, the next study, a larger study, perhaps in partnership with government like the NIH, or maybe even getting an industry partner, but also in helping to protect the results through the development of patents and maybe even spinning out biotech companies, and also to help design the new formulations or the new combinations of drugs that would enable the commercialization strategy and market access and ultimately patient access to these drugs. So one of the advantages of repurposing is that the commercialization strategies we've been talking about today could take years, but the advantage of it for now is that these drugs are available and they can be prescribed for patients. And I personally do prescribe drugs off-label because I feel the data is so compelling and these are safe drugs for patients. But I would say to patients and their families that just like I've been asked by many of my patients and families, what can I try? We want to try something. I think you can express that to your doctor and even present them with the results. Dr. Giacomo's results have been reported in the Journal of American Medical Association, and Dr. Pereira's results have been submitted for publication and hopefully will soon be available to the practicing physician world in the near future. So with that perspective, Anna, I was just wondering, given the data that you've seen, if in your patient population that you're treating as a neurologist, if you're being asked about off-label usage, and if you're prescribing, for example, riluzole for your patients at this time.
I don't prescribe off-label. I usually encourage patients to go to clinical trials, and I think the reason for that is I would like to see. I think those data, the phase 2 trials are promising, but they are not conclusive, and we really need to get to a phase 3 trial to really prove efficacy and safety more in a new population in a more profound way. So since we really need for us to move the field to really get to new drugs, we need strong data, and not only my trial, but multiple other trials. And I think this is the only way we can get reliable data for novel therapies that could be disease modifying, meaning slow down the progression of the disease or many other aspects of that. So in terms of this, also the other reason for clinical trials is to have a more safety monitoring. Riluzole, for example, have to monitor for blood, for liver tests and other things, and white blood cells. So having a structured environment that can provide more safety control for patients as well. In terms of having those drugs running to phase 3 and getting FDA approvals, I think, as Howard pointed out, we chose a very tough field, right? So Alzheimer's is really tough. It's not like a skin disease. In 2 weeks, you go there and check the skin, is it better or not? So you have to check how those patients do over a year in terms of Alzheimer's advocates, NIH, philanthropy, and ADDF has played a major role in changing Alzheimer's in the last 5 years. We have a lot more funds to be able to understand the mechanism of the disease, come up with more targets that are validated to test out in patients.
Giacomo, so let me ask, I'm wondering if you prescribe patients off-label drugs, if you get asked by patients about taking off-label drugs, how you advise them for clinical trials, and then again, the second question is, how long do you think it'll be before rotigotine might actually get some sort of FDA approval?
Patients started asking me whether they should also include rotigotine in their therapeutic scheme as an add-on to standard of care. And my advice is always to try. And this is actually somewhat problematic in terms of reimbursement from the National Health System, because the indication is for Parkinson. Some patients can afford this expense, some other cannot. So maybe this is somewhat concern. That's why we want to move further, and we want to demonstrate that this drug can be effective and make it available to everyone. Of course, we have been testing this in a relatively small sample of patients, so probably there will be also some patients that will respond better than others. And if we enlarge our population and we perform a study with biomarkers, we probably will be also able to say in the future to our patients, okay, for you this drug can be effective, and maybe for you won't help very much, which is probably the reality. Having these patients, it's also very delicate in terms of providing some hope. So every time we have some good news or something that is new for them, it's important, because you are adding a small piece of hope. We all know that nobody is claiming that it is going to be the cure for Alzheimer's disease, but we have to be very precise and describe how this new piece of evidence may slightly improve their life. We don't have in Italy the opportunity to work with foundations such as the ADDF, and so by my side I'm really appreciating the great effort that you're doing in the States by not only funding studies in the US but also outside the country. To answer to the second question, I think that if we are going fast and we try to perform a phase 3 clinical trial as soon as possible, maybe in 3 to 5 years we could go to the FDA and provide solid data sufficient to get approval, obviously if we are lucky enough to confirm the data that we got in the phase 2 trial. I would like to ask Howard, in the first part of this conversation, he was mentioning that ADDF is turning his interest from preclinical studies to the clinical ground. So I would be very interested in understanding how in the future, Howard, you see the combination of these two important side of the coin, if they can still work together, or how do you see this interaction, which is very important?
Historically, going back to the beginning of the foundation back in 1998, when the science wasn't as advanced, I'd say 80-85% of our portfolio was preclinical science. And we probably funded something like 500 preclinical drug discovery programs over the last 22 years in 19 countries. And so there was really a strong feeling that the need had shifted, because also agencies like the National Institute on Aging has $3 billion now to spend every year on preclinical science and other things like clinical trials. You know, that the need isn't as great for preclinical risk-taking. So now our portfolio is reversed, and now it's 20% preclinical and 80% clinical. So we haven't given up on preclinical, we've just shifted our emphasis. There's over 120 drugs in development, and we have our fingerprints on about 20% of those. We want to help to advance these clinical trial programs that are risky. They don't have a lot of commercial interest unless we help them to develop. And so, you know, agencies and so on aren't necessarily investing in repurposing. And somewhere about half of our clinical trial portfolio is repurposing, which we think we can learn a tremendous amount. But ultimately, I think our strategy is based on the fact that Alzheimer's disease is a uniquely human disease, really. If you think about it, I like to joke rather seriously that mice don't write books. You know, and there's elements that we've talked about today in terms of executive function that, you know, mice don't just have executive function, or at least as far as we know, they don't. So I think that combining the fact that we have so many targets in development now, we have so many proof of concept in animals that haven't been tested in humans, and that, you know, really the ultimate test of any drug, especially in Alzheimer's disease, has to be in humans. I want to thank Giacomo and Anna for participating with us and for contributing their thoughts to our day. And if you have any questions, we are going to have a live Q&A And so we'll look forward to hearing your questions in the audience in our next session.
I'd like to welcome everybody to our live question and answer period. If you have any questions, please do send them along to us in the chat, or you can send them. So I just wanted to begin with our first question from the audience. And what is the next step in the trials? Anna, you kind of talked about it, but I guess, your study should be completed and all the data analyzed quite soon. And I wonder how you're already starting to plan, because I know you've seen the results. How are you planning for the next study, and where do you think the funds will come from?
Thanks for the question, Howard. I think related to the pilot trial, so the trial has been completed, the data has been analyzed, and it's on the peer review right now. Just to highlight, so our key finding is that we saw that there was less decline in cerebral metabolism in patients who were taking riluzole rather than placebo, and that correlated with cognitive performance. And FDG PAT was the biomarker we used, is one of the best predictors in terms of biomarkers of progression of disease and cognitive decline. So there was a pilot study. We enrolled 50, we screened almost 100 patients and enrolled 50 and completed 42. So now the next step is to really look into that in the phase 3 trial, which would give us more definitive answers for both safety and efficacy. So I think, as I alluded before, we have 2 strategies ongoing. One is to use exactly the same drug, the same dose in a phase 3 trial to already have some proof of concept, of efficacy in the human brain. And then we're going to start looking into both NIH funds and as well as potentially private partners, since phase 3 trials in Alzheimer's are extremely expensive, require lots of people, very long studies, and neuroimaging, which are quite expensive. And the other strategy that we have is a new formulation of riluzole in a way that you can diminish potential side effects from the current dose, and improved compliance with the once-a-day medication, less likelihood of liver issues, better absorptions, and things like that. So that's going to take a little longer because we're still developing the molecule and things like that. So we have one is a faster track path, and the second is a little bit more improved molecule that would segue to the first one. So.
And could you a little bit more detailed describe what you think some of the clinical benefits that you saw in the trial were of riluzole on either cognition or function, for example?
Yeah, so it's interesting that the most robust finding we had from the PET scan data, I think as I mentioned before, PET scan measures glucose uptake in the brain. It's really run by glucose. So if there is a neuron that is sick, that is not functioning well, you uptake less glucose. So the area that saw a most robust effect on less glucose, the reduction, is called the posterior cingulate, which is a key for me as a cognitive neurologist, all about networks in the brain and how that leads to cognition. So the posterior cingulate is one of the areas of the brain that is earliest and most affected in Alzheimer's disease. It's a key hub network that connects to multiple areas of the brain. So the posterior cingulate, as well as multiple other brain areas, including the temporal cortex, is important for memory, for language, semantic memory, and as well as we saw frontal and orbital frontal cortex, which are areas of the brain related to behavior as well as executive functioning.
So did you see those improvements in behavior or in memory, or what actually in terms of the clinical effects did you see?
Yeah, so we are powered primarily for FDG PAT. So those FDG PAT measures correlated very well with cognition. As well as I would like to point out that the FDG PAT, so we correlated very well with ADAS-Cog, which is a measure that is used for Alzheimer's disease. which is a composite measures of many things, language, memory, and things like that. With NPI, which is NPI is called
neuropsychiatric interview.
Yes, and so it's very important for us clinicians. We see that one of the major challenges of Alzheimer's patients is the behavior, agitation, anxiety, hallucination. So we had very strong correlations with NPI, and if you analyze the data as a single analysis, NPI had a very, very good trend there, as well as ADL. ADL is called activities of daily living, which is a real functionality measure.
So in summary, I guess what I would try to just sum up is that you used a biomarker PET, which measured glucose, which is energy to the brain, and you were able to analyze it, I believe, with one of the companies that we funded, Dawn Matthews. We funded her company several years ago to help you do those analytics on the PET scan. And that when you saw improved glucose uptake on the PET scan in specific areas of the brain, that correlated with some of the clinical findings that you discovered in the clinical trial, that there was improvements on cognition in those specific areas of the brain that are responsible for those cognitive effects. And that's very exciting, and it illustrates how biomarkers and clinical assessments of cognition can be used together. Giacomo, your drug, we were particularly excited about your drug because we talk about executive function and how disabling it is, and that it's apparent very early in the disease, even in the earliest phases of mild cognitive impairment. But it's very hard to measure, and there are no drugs that we know of that really improve executive function, which is the highest order of thinking that human beings conduct. Could you tell us a little bit more about what is executive function exactly? What kind of behaviors would, for example, the lay people in our audience look for in the earliest phases of MCI or mild dementia that would be reflective of executive function? And how did you measure executive function in your clinical trial?
Thanks, Howard. That's a very good question. So executive functions are one of the most important features of our frontal lobe activity. And by the term executive functions, actually, as cognitive neurologists, we mean a set of skills that are very important in everyday life activity. Namely, decision-making processes are under the umbrella of executive function. The ability to predict or to estimate the passage of time, the ability of keeping memory few issues for a certain amount of time, such a telephone number. This is more related to working memory, but it's also under the umbrella of the executive function. And more importantly, I think it's the control that the frontal lobe has on everyday life activity. That's why we think that this may have a serious impact in Alzheimer's disease patients' everyday life.
Yeah, so I mean, the way I see it in my practice very often is people come in and they talk about apathy, that patients, that used to be active playing golf. I remember one patient in retirement, he used to play a lot of golf, and he'd plan his golf trip for Tuesday or Wednesday at 1 o'clock, and then he'd call his friends and say, you want to play, a foursome at 1 or 2 o'clock, and then he'd drive there and he'd go play. And now his wife is complaining that he doesn't play golf anymore, and she wants to know if he's depressed. But the problem is that he can't do planning anymore, right? So he can't actually put together the cognitive tasks that are necessary to think about Tuesday, to think about what he wants to do on play golf, to make the phone call to make the reservation, to call his friends, to get there on time, because he has to remember to get there on time. And it becomes even more serious than that. As you mentioned, emotional control is also a cognitive function that people lose. So very often, lay people will describe executive dysfunction as a loss of emotional control. Somebody who used to be very passive, for example, might be getting easily angered. And it's often referred to as an early symptom is change in personality, which is a reflection, really, I think, of a lay term that's reflecting this executive dysfunction. And multitasking, right? So people lose the ability to do multitasking, which is also a frontal lobe function and executive dysfunction. So these are really serious disabling events, and rotigotine seemed to improve them, and so that's why I think the rotigotine study is going to be so important in people's lives. And you were actually able to show that not only did you see improvements in cognition, but you actually, to make them real world improvements and not just findings on a cognitive test, you saw improvement in function, sort of the way I was just speaking in terms of the ability to play golf, for example. So that's very important. Sort of extending the theme of biomarkers, which I know you also used in your trial, Giacomo. What do you think about this new blood test that is on the market from C2N? It's a plasma A beta test, and there are other blood tests that we've heard about. The plasma tau test. There's 3 of them out there now, and we're seeing the excitement about other blood tests. But the one that's on the market for plasma A beta, how would you think about using that in your clinical trials? And how do you see that, ultimately affecting perhaps your clinical practice?
Yeah, I think that's a terrific opportunity we have now, because so far, Anna was mentioning FDG PET is very sensible, but can be costly. And by measuring E-beta levels and tau levels so far, we were constrained to lumbar puncture, which obviously can be done once and not repeated so easily. But now we have the chance to measure A-beta levels in the blood, and as you mentioned, hopefully also some tau phosphorylated proteins seems to be a reliable measure in the blood. So the scenario is becoming extremely interesting, because we want biomarkers, we want reliable biomarkers, and I think that we want also to combine different levels of biomarkers, namely the blood levels of the underlying beta and tau pathology, but also we want to see what happens in the brain by using FDG PET, as I was mentioning, but also by using, for instance, in our case, transcranial magnetic stimulation in combination with EEG, in order to see effective changes in cortical activity and connectivity. So I think this is really fascinating, because it's adding and increasing the threshold and the levels of our clinical trials that are not only to provide a clinical endpoint which is relevant, but we can learn a lot more about the underlying mechanisms.
That's very interesting. Go ahead.
Oh, thanks, Hon. I would add, amazing, Giacomo, is that I think it's going to really facilitate a lot screening for clinical trials, because we, as cognitive neurologists, see patients coming with different cognitive syndromes with different underlying neurodegenerative disorder. So we can screen patients much better, much more easily, much cheaper. And then, since brain imaging can give you neuroanatomical localization, we can decide which patients are going to go for brain imaging. But I think it's being a breakthrough in the field to be able to diagnose and follow these patients longitudinally in a much easier and much less expensive way.
Yeah, absolutely.
Yeah. And it's a real breakthrough, and it correlates with the PET amyloid scan. So whereas the PET amyloid scan right now, if I order it in my practice here in Manhattan, it's going to cost $5,000, and basically nobody pays for it. None of the insurers, Medicare won't pay for it. So it's not widely used, but I find it extremely useful. I just had a patient last week who we all thought had Alzheimer's disease, and her amyloid scan was completely negative, which was really helpful in understanding what her disease was. It looks like it's more of a microvascular disease of her brain, and maybe has a different prognosis. And it was just a different approach to treatment. We'll get her to exercise more, knowing it's a vascular disease, and manage her hypertension much more carefully than she's been doing over the years. And I think it was very comforting to her to know that it wasn't Alzheimer's disease. I was asked as our last question, I guess, about some other things that ADDF is doing that's exciting, and I'll just really quickly mention three studies. One is another repurposing study using a Parkinson's drug called rasagiline. This was just reported out in the last quarter. Again, positive findings, exploratory study that's exciting enough for Dr. Jeff Cummings at the University of Nevada in Las Vegas to seek to do a phase 3 trial, and we're going to figure out a commercialization strategy for that drug. He got really interesting results with that drug, and it could be one that could be repurposed. Another company called Amylyx, which we funded to do a combination therapy. And as I've often said, we're going to need combination therapies for Alzheimer's disease because there's so many different pathways to neurodegeneration in Alzheimer's disease. And this Amylyx trial, which recently using the same combination of two drugs, showed efficacy in ALS, which is a really hard disease to manage, to treat, that we're very excited that in the next quarter, we're going to hear results on the Amylyx combination therapy, which I'm really excited about. And then lastly, we talked about a little bit about new chemical entities, and we haven't really spoken much about it today, but new drugs that are completely different mechanisms and exciting. And I think the one that's coming up on the horizon that we're all really hopeful about, and the data are just about completed, and should be hearing about it at some of the upcoming meetings, is pharmatropics, which we've been funding about 20 years now. And the phase 2 trial is just about to report out in the coming month or two, I believe. And if that works, it'll really be a big breakthrough, I believe. So I'm very hopeful on that. With that, I want to thank all of you for joining us today, and particularly being part of this question and answer. I want to mention that this has been, even though virtual, which who would have thought, is our most successful Palm Beach event today. And I want to particularly thank our ADDF co-founder, Leonard Lauder, who doubled his donation this year, helping us to raise over $800,000 for Alzheimer's drug research. Thank you so much for Leonard. Thank you so much. And thank you to all of you who are on the line, who have donated and contributed in so many different ways to progress of ADDF. None of the work that you heard today would be possible without your help. And we're really, really grateful to all of you for being our partners in discovering new drugs for Alzheimer's disease and achieving our mission of one day having a cure, hopefully in our lifetime. Thank you so much.
And now, ladies and gentlemen, here's Paula.
Thank you so much.
Good morning. Paula's here. Yes.
Happy to be here.
Howdy.
We're off and running.
Apparently, when I was in preschool, 3 or 4, one day we went into class and everyone was going around in a circle sharing what their parents did. And one kid would say, my mom's a doctor, and another would say, my mom's a lawyer, and my mom's an artist. And they got to me and I said, well, my mom works in a box. And everyone was very confused. I think it was only a little bit later that my understanding of what she did grew a bit more sophisticated.
And I don't know how many of you have gone through Army or Marine or Navy training, but it is extremely intimidating.
We all take for granted these days the media personalities we see, particularly on television. I remember those early waves of females breaking through the glass ceiling. The names like Katie Couric, Diane Sawyer, Paula Zahn.
Are you saying he doesn't have the capacity
I'm
to serve
not
as
saying
president?
anything, and I've told you, Paula, that you are a great sleuth, one of the great investigators here, or what is it? What do you call it when you pull information out of a guy that doesn't want to give it, you know? Not inquisition.
Thank you. So I have been privileged to witness history from a front row seat and to bring it into your homes. Fall is becoming reality as Germany enters a new era. How can it be that hundreds and hundreds of thousands of victims have not received any food and water more than 100 hours after Katrina hit?
On 9/11, it was incredibly scary after the attacks in New York and over the country. I remember her going out of her way to pick me up from school, make sure that I felt safe, brought me home, and then she actually went into work that day at CNN, and it was her first day on the job.
I was stuck in Europe, and it was very reassuring to hear her and to see somebody that one trusted.
At this time in the history of this country, they needed a person who was respected, who had journalistic experience, whom they could put in the air live, who could handle everything, and yet could also give a sense of calmness.
You're a terrific journalist, a total pro, class act. Got nothing but the greatest respect for you.
Despite her very youthful appearance and energy, she's been doing this for a long time. She has shown versatility. She has shown consistency. She has shown a professionalism that's gone from, media niche to media niche to media niche across decades. Paula Zahn, media pioneer, media icon.
How do you describe somebody that is just so multifaceted and so multi-talented in so many ways?
Would you please welcome my co-worker, her debut tonight at Carnegie Hall, Paula Zahn.
I grew up in a very musical family. My mother was a pianist. Among my siblings, we had a clarinetist, a violinist, a percussionist. But I truly fell in love with the cello. My brothers played competitive football and rugby and basketball and baseball, and if my sister and I didn't get into sports, we would have been left behind by them.
Paula Zahn participating in the Pro-Am, the only woman that has ever been invited to play in this.
Golf is something we share in common. She played in college and was a scratch golfer, and to this day she plays maybe once a year, if not once every other year. And she's just smacking the ball past me with the driver and scoring really well, and it's just incredibly frustrating to watch.
Well, here's a duck down here. Did you eat my ball?
To test your abilities, I think we got to get in the gates. We can have a little grudge match right here.
Woohoo!
Good!
Obviously, our mom had a super-powered career and she was immensely busy. And I think one thing that I have always really appreciated and admired about her is how much she made us feel prioritized despite all of the other important things that were on her plate. And she would never miss a sports game, no matter how bad, my basketball defense skills were.
They were bad.
They were bad.
Paula is the best sister imaginable. Family is everything to her.
She is unfailingly loyal and devoted to her family and friends. I couldn't be more proud of the woman and sister and person that she is.
And then personally, Paula's been so directly touched by Alzheimer's.
My mother was diagnosed with Alzheimer's 10 years ago. Sadly, my mom hasn't recognized any of us in 7 years, and we were her life.
Watching our mother, who was so vibrant and intelligent, mentally decline was absolutely heart-wrenching.
She would, read 1,000-page history tomes for fun and finish them in lightning fast speeds. She taught herself to paint and filled, all of our walls with beautiful watercolors.
Paula took on her disease being the main advocate because she lived in New York, whereas the rest of us lived thousands of miles away. She has given us a peace of mind that our mother is being loved and taken care of.
Leonard and Ronald Lauder, who always had a special eye for talent and exceptional people. They co-opted Paula into joining the ADDF family, and she not only has joined it, but it has become, like everything else she does in her life, a true passion.
I think Paula Zahn's the perfect honoree for our Palm Beach luncheon.
No one deserves this award more than Paula. She's the unsung hero who's always there for every event, in hosting and being the face of ADDF at every function.
So Paula, thank you. You are a star, and thank you for today.
Paula, we are so excited that you are the honoree for the Hope is on the Horizon Award for ADDF. You are just the best.
And Paula, so say I, and so say all of us, there's no one like you, and you set the standard for all of us to follow, which is love of family, love of the family, and love for the future. You've helped the future of your family and all of ours by doing what you're doing. So thank you for that.
Much love.
Congrats on your award, sis. Your work with ADDF has such meaning for our family, and I am just so proud of you.
Congratulations, big sis. I could not be more proud of you and the work you have done for ADDF.
Congratulations, Mom. We love you so much. We're so proud of you for everything that you do, and are thrilled to see you receive this honor.
We couldn't be prouder, and the ADDF couldn't be luckier to have you behind this cause.
Oh, Paula, how fortunate are we, the ADDF, to consider you part of our family. Thank you for the years of passion. Thank you for the years of philanthropy. Thank you for the years of service. Paula, your entire ADDF family, we salute you and we love you.
Thank you, thank you, thank you for this wonderful award. I am deeply gratified that ADDF is recognizing my work for this critically important cause, a cause which I know is very near and dear to many of you out there. I'm hopeful and perhaps I should say confident that the work of the ADDF will someday lead to a treatment for Alzheimer's and perhaps even a cure. And it is a privilege to have joined this organization in the fight against this horrible, horrible disease.
We hope you will consider making a donation by texting ADDF to 243725. And remember, because of the generosity of the Lauder family, 100% of your donation goes directly toward the science. Thank you to each and every one of you again for making today possible. At the ADDF, we have a true north. It's the patients, the caretakers, the families. It's the scientists. It's the innovation. It's how we go from bench to bedside, turning hope into reality. To everyone who participated today, to our incredible chairs and co-chairs, to each and every person that appeared in these videos today, to Paula as our honoree. We could do none of this without you. We are indebted, and we hope you'll be back next year when we can be in person. Stay safe and healthy, and thank you for joining us today.